So in today's lecture, we're going to talk about DNA technology, and we're specifically going to talk about bioengineering, making genetically modified organisms, genomics, and things like that. So DNA technology has allowed for lots of very important scientific advances. And so just to name a couple, there are now crops, so crop plants that have been genetically modified and what the modification has been is that they can produce their own insecticides. So instead of having to spray pesticides onto crops, the crop plants actually have a built-in insecticide inside of their cells, and so insects can't eat it without getting sick, and it's not dangerous to us because of the insecticide only works on insects. And so this is something that's happened with a lot of crops. Another big thing that's been one of these big advancements is just being able to look at human genes. Okay, so not only can we compare human genes to one another, so in kind of a DNA fingerprinting, which is used a lot in crime, kind of examining crimes and trying to see if the DNA that they found at a crime scene matches a specific suspect, but also in the evolution of humans. So we've been able to look at not only living humans, but extinct human relatives like Neanderthals, and trying to compare the genomes to each other. We also can do this with other organisms like chimpanzees, and so we'll talk a little bit about that. There has also been significant advances, in detecting and curing fatal diseases. So we know many diseases are caused by defective genes, and so if we can detect those genes, we can screen people for those genes. We also can then maybe go in there and fix these diseases. All right. So a lot of what we're going to be talking about today, we're going to say different terms like biotechnology, and genetic engineering. Okay, so I think both of these you can genetic engineering would be under something like biotechnology. So biotechnology can be kind of the umbrella term, and then genetic engineering is going to be a specific type of biotechnology. Okay, and so we'll talk about some specific types of biotechnology that we have done. Okay, so biotech. Okay, so when we're doing biotechnology, what's happening is that you're manipulating organisms or parts of organisms to make some type of product. So one of the things we're going to talk about is using bacteria to make insulin today. Okay? This idea of manipulating organisms is very, very old. Okay? And so this is going to date back to the dawn of civilization. Okay, so when we started farming, when we started kind of domestication of animals, those types of things can be considered kind of manipulating organisms. So you decide to plant specific types of food that you enjoy eating. So many kind of ancestral species of plants didn't taste very good or were poisonous. And so over time, humans have selected the ones that tasted the best that weren't poisonous. And those are a lot of the varieties that we see today in our fruits and vegetables. We've been kind of using yeast for our bread and beer for a very long time. So once we started being able to farm and grow wheat, beer and bread, so again, this is bread, were things that we were using. And we're specifically harnessing yeast to help make both bread and beer. Okay, and then again, breeding livestock, specific types that we want. Dogs, we domesticated dogs. We selected them for things that we want. Today, when we think of biotech, it's more sophisticated and it uses kind of groundbreaking DNA lab techniques. Okay, so we're not talking about just planting the seeds of the plants that you want to eat. We're now going to talk about specific stretches of DNA, selecting parts of the DNA, and actually putting them into other organisms. Okay? And so things that we can do today are we can modify genes and move them between organisms. Okay, and so we're going to talk about how that actually happens. So there's a lot of terminology when it comes to this kind of DNA technology, and so I'm going to be talking about specific different terms right now. And so this first term, and this is probably something that you've either heard or have seen on food packages, and this is the idea of genetically modified organisms, or GMOs. Okay, and so what a GMO, or a genetically modified organism, is defined as an organism that has acquired one or more genes by artificial means. So that means that humans are putting these different genes into other organisms, okay? There's also a different term called a transgenic organism. Okay, and so if the genes that have been acquired within an organism are from another species or more than one species, they're considered transgenic. Okay, so I'll write this down. So if the newly acquired genes gene is from another organism, and this is typically a different species. So you wouldn't be like human to human. This would be like giving a human gene to bacteria, typically of another species. The recombinant organism is called transgenic. So again, the big thing is that it has to be from another species, usually for it to be called transgenic, okay? So species, like humans are species. There are no other species of humans, but like, you know, we have multiple different species of birds and flowers and all that kind of stuff. So that is kind of a classification of relatedness. And so this means that it'd be from a different species. So not within your own closely, your species. Okay, this word recombinant is important. So recombinant is recombining DNA. So we've talked about recombinant DNA in kind of crossing over and other kind of mechanisms. So recombinant DNA is going to be when we're kind of putting more DNA together from different sources. So recombinant DNA. Okay, so this is going to be combining pieces of DNA from two different sources and again, this is also often different species to form a single DNA molecule. Okay, so we're going to talk about how to actually do this. So you'd be taking a sequence of DNA and then inserting that into, you know, let's just say we have this orange segment from a different species. We're inserting it so it's actually part of the DNA molecule at this point. So it's not just kind of separate, it is actually incorporated within the DNA molecule. Okay, so the next term is genetic engineering. Okay, and so this is going to be the direct manipulation of genes for practical purposes. Okay, so and let me just say that there are also is going to be some type of genetic engineering that's not necessarily practical. So for example, fluorescent genes from jellyfish have been inserted into fish, and you can buy these fish and they kind of glow in the dark. That doesn't really have any practical purpose other than making people happy and want to own them. But the stuff that we're going to talk about today is going to be for practical purposes. And so, for example, one of the big things that happens is that there are genetically engineered bacteria that mass produce chemicals. Okay, so we have taken bacteria, inserted genes that we want to make some type of chemical, and some of these things are going to be cancer drugs, other types of drugs, pesticides are another type. Okay, and so this is using bacteria as basically little factories to make different things that we want. Okay, and there have been times where there's been transfer of genes from bacteria to plants. Oops. And from one animal to another. Okay, so all of these are types of genetic engineering where you're manipulating the genes for a practical purpose. So one example for animal to animal trying to think if there is one. We sometimes would put like human genes into other type of organisms to then look at specific things that are going on, maybe with different health issues. Okay, so we're now going to actually talk about how to do this recombinant DNA. So we're going to talk about recombinant DNA techniques. Okay, so bacteria are extremely important in this. So they are the workhorses of modern biotechnology. Okay, and one way that they are very useful is that they have something called a plasmid. And so we're going to talk about these plasmids. Okay, and so we can manipulate genes using bacterial plasmids. Okay, so plasmids are an important term. Okay, so what are plasmids? Plasmids are going to be kind of short, small circular DNA. Okay, so small circular DNA that duplicates separate from the larger bacterial chromosome. Okay, so there is, if you have a bacteria and they're going to have lots of DNA, it could be all kind of connected. There are these circular that are not connected to the normal DNA, and these things are going to be the plasmids, okay? And when the process of replication happens, they do not combine with the other DNA. They stay separate by themselves. And so when you go through DNA replication, you then end up having, two plasmids at the end. So these plasmids are really, really important, okay? And so what can happen is that you can actually put genes onto the plasmids, okay? So you can carry virtually any gene on the plasmid, and they would be passed on from generation to generation. Okay, so if you can get a DNA molecule, a gene connected to a plasmid, so let's just say that the black is representing the bacterial DNA, the plasmid, and then this orange section is going to be whatever gene we're interested in. If we can get the gene inserted into the plasmid, we can then make tons of copies of this, okay? So these are really important, and specifically they're important in something called gene cloning, which is basically just making many, many copies of a specific gene, okay? So this would be projection of multiple identical copies of gene-carrying DNA. Okay, and so what that gene carrying mean is that this gene right here would be translated into some type of protein or some other type of product. Okay, so it has specific product that's going to be associated with it. All right, so how does this actually happen? Okay, and so first I just kind of want to show you a better picture of how this looks. And so this would be our gene of interest. So this could be a human gene, for example. This could be an insulin gene. And this is the plasmid. And so we'll explain kind of how the plasmid opens. Originally, the plasmid will be connected, it'll be a full circle. But if you can cut it open and then you can insert a gene right there. And so then this is what we would then call recombinant DNA. So then if this recombinant DNA is replicated over and over and again, we're going to have many, many copies of the gene that we want. And so we're basically trying to make as many copies of this gene as possible. Okay, so let's talk a little bit about this process of how to clone a gene. Okay, and a clone is just a copy. All right, so the example we're going to talk about is going to be, so we're going to do DNA cloning to synthesize a protein. And this protein specifically is going to be insulin. Okay, and this is going to happen in bacteria. So we're going to take a bacterial plasmid and we're going to put a protein that is going to code for insulin. Or we're going to put in the DNA sequence that's going to code for the protein insulin. All right, so the first step of this process is that you actually have to cut open the plasmid, and then you paste in the gene. Okay, now this process requires something called restriction enzymes to actually cut open the plasmids. And so this week's lab, we talked about restriction enzymes. So let me just write this process relies on restriction enzymes. Okay, and so restriction enzymes basically will cut the DNA. So they make a double-stranded cut, okay, and then the DNA will basically be open. So restrictions, and actually let me show you this other image. This is better. So initially you have just the plasmid. It has nothing in it that is not, supposed to be there initially. And then what happens is you use something called the restriction enzyme. The restriction enzyme comes in here and cuts the actual plasmid open. We then can use this target gene that we want to insert, and it can be put in with this plasmid that's been cut open. And then we do something next, which is called DNA ligase, which then will kind of cut and paste basically everything back together. Okay, so the restriction enzyme cuts the DNA, and then we have something called DNA ligase that is going to join DNA together. Okay, so it acts kind of like a glue to put the DNA back together. All right, so that is kind of the first step. And here's just another little diagram about what restriction enzymes are. So the main thing you need to know is restriction enzymes are going to cut the DNA, okay? They do this because they have a very specific sequence that they are able to find. So the restriction enzyme, which is this green thing, has kind of a way to detect specific sequences of DNA. So for example, this restriction enzyme is recognizing this sequence of DNA. So it's GAATTC. Okay, so it's, obviously they go both directions. So when it comes into contact with this specific sequence, what it's going to do is actually going to cut the DNA. Okay, so the DNA will then be cut, and you then have kind of, the way it cuts it is it has this little bit of an overhang. So they can then join back together if you have another piece of DNA that you'd be interested in putting it there. So the main thing is that it's going to cut the DNA. Ligase will then come in here and join everything back together so that you have no gaps. Okay. So the next step, so after you have the plasmid with the actual, actually, let me go back to this page since I still have room. So our next step is going to be to insert the plasmid into bacteria. Okay, so when we have the plasmid originally it is not in the bacteria, so we've kind of taken it out of the bacteria. We've then done this copy paste, or cut and paste with the first step. And then the plasmid has to go back into the bacteria because the bacteria is going to have all the necessary ingredients for kind of replication to take place. Okay. Now, an interesting thing that happens when they do this is that they use an antibiotic to identify which bacteria have actually taken up the plasmid. Took up plasmid. Okay. This step of plasmid being taken into the bacteria is called transformation. And the way that the transformation takes place is you have your plasmids that have this DNA that you want to be in there, okay? You then do something called a heat shock, which means you basically really heat up the bacteria. And I looked into it, and they don't actually know why this happens, but when you heat shock bacteria, they're more likely to kind of have that plasmid taken up into the cell. So what I want you to see is that there's been a heat shock, and then we have 4 bacteria right here. Only one of the bacteria has actually taken up the plasmid. And so when we're looking for bacteria that have this plasmid, we need to be able to somehow identify which ones have it and which ones don't. Now, one thing that I didn't mention before, I kind of skipped over this, is that one of the things that the plasmid has within it is this red little sequence, and this is an antibiotic resistance gene. So what this does is when they have the bacteria, they've done this process of trying to insert the plasmid into the bacteria, they can then put the bacteria on a plate with growth media, but they also can treat the plate with antibiotics. And so only the bacteria that have the actual plasmid inside of them will be able to survive, okay? Because that antibiotic will then kill the rest of the bacteria. So if we're looking back on this page, because only this bacteria has the antibiotic resistance gene, these 3 would all die because the antibiotic would kill them, and then only this one would survive. So once you've actually used the antibiotic, you can select the colonies that have survived, and those are the ones you're going to then want to culture, where it just basically means make a lot of them, okay? So let's see. Okay, so let me I'll write that down. So this is part 2 continued. Okay, so the plasmid carries antibiotic resistance gene. Okay, so bacteria that didn't take in plasmid will die, and the ones with the plasmid survive. Okay, and then the third step, what you do is you take those colonies that survive and grow up lots of plasmid carrying bacteria. Okay, and they basically act as factories. okay, that are going to make protein, whatever you're interested in, okay? So once you have the colonies that we know have the plasmids, they can then be put into growth media, and this is basically just going to be some type of food source for the bacteria. You can actually have massive vats of these. Sometimes they look very similar to how kind of beer is brewed, like these massive metal containers. And you just have just tons and tons of bacteria. And then you can, if you put them in the right conditions, they'll start to express the gene. And so if we're looking for specifically, you know, this blue gene that we had, and it's going to turn into insulin, well, once the gene is transcribed, it can then be translated, and then these little red things are going to be the protein that we're interested in, insulin. Okay, so this process is something that happens a lot and is used, you know, quite a bit. All right. Okay. So one thing I wanted to mention is kind of this idea. So right now we've talked about how to make large quantities of proteins, but there's also this idea of being able to do gene editing. Okay, so not necessarily having bacteria take in a gene, but to actually be able to go into a cell and be able to manipulate the gene, okay? And so one of the biggest, you might have probably heard of this on the news, is this kind of system called CRISPR, Cas9. Okay, so this is a system of gene editing, okay? And so this is sort of a complex thing. This is something that you're probably gonna hear about a lot, because it's one of these huge, big technological advances that are discoveries, I guess you could say. And so what CRISPR allows for is, allows the nucleotide sequence of specific genes to be edited in living cells. Okay, so the idea is that if we could get this to work in humans, we could go in and remove mutated genes and replace them with normal genes. Okay. So if you have a faulty gene, for example, we've talked about sickle cell anemia, so there's been a mutation in one of the bases. If we could use CRISPR, we could then have it go in there and fix that mutation so that those individuals would start producing the normal hemoglobin, okay? Obviously, that is not something where we're at, but that's kind of where this type of technology is going. Okay. Now, the main way that this is going to work is it also uses restriction enzymes, and I'm going to just write restriction enzymes as RE. Okay. And so what restriction enzymes, they guide to specific locations in the genome by specifically designed RNA molecules. Okay, and so these molecules will then kind of move through, and whatever sequence they're going to match up with, they will then bind to, and we'll be able to cut and basically make that change. Okay, so this is about all I want you to know about CRISPR. It's pretty controversial topic right now about like who owns the rights behind CRISPR, should it be able to be owned and things like that. But this is kind of where the future of medicine, they think, is going, where you would then be able to go into an individual that has some type of mutation and fix it for them. Okay. Now, medical applications are one of the biggest things that a lot of genetic engineering is trying to kind of deal with. So I just want to talk about a couple different medical applications. Okay, and so one of the medical applications is that you can, again, do this type of plasmid work where you can put a desired protein into bacteria, and then the bacteria, or it can actually be a different organism, it could be something like yeast, can produce large quantities of those proteins. Now, one example, a real example, and I've kind of already talked about insulin, but this idea of humulin. Okay, so this is a human insulin produced by genetically modified, and I'm going to put GM, genetically modified bacteria. Okay, so previously, when modern medicine was trying to treat people with diabetes, they would use insulin from cows or pigs. Okay, so let me just write that. So insulin for diabetics was previously derived from cows and pigs. So taking cow and pig insulin and then using that itself. There's lots of problems with that because they are not humans. There are some compatibility issues. So that was not ideal. But then in 1978, scientists actually inserted the two genes that code for insulin into E. coli. So not all E. coli is dangerous, so it's something that's used quite often in laboratories. So this is the kind that would not make you sick. The E. coli had the two genes put in there, and then that was in 1978, and then in 1982, so not too many years after that, humulin was the first genetically engineered pharmaceutical product. Okay, so humulin becomes the first genetically engineered pharmaceutical. Okay, that was approved by the FDA. Today, there are more different types of proteins and things like that are produced. So human growth hormone, this is human growth hormone, is one of them. There is a protein that dissolves blood clots. That's one of them. Vaccines are often going to be made this way. And then EPO, which is kind of helps make red blood cells for individuals that are anemic. That is also something that's made in this form. So they insert the genes into bacteria and then are going to basically use them as these factories. So moving on from there, so these are medical applications, but lots of genetically modified organisms that we talk about, especially in the news, come from kind of agriculture. So we're going to talk about genetically modified agriculture. So GM agriculture. Okay. And so DNA technology, the type that we're talking about today, and not just kind of selective breeding, has replaced how we used to breed our different crops and also our different animals that we have, okay? So DNA technology is replacing traditional breeding programs. Okay, not all. So there are people that breed specific types of animals that do not use genetically modified or DNA technology. So at this time, in the US, almost all corn, soybean, and cotton are genetically modified. Okay, many times they're going to have kind of insect resistant plants. Okay, that so they have these insect resistance. So basically they make the plants are the chemicals needed for insecticides. Okay, which then this reduces additional chemical insecticides. Okay, all of these types of things have been tested and they have found no kind of issue with human health, because oftentimes the way that you would kill the insect is in a way that is not biologically relevant to a human. So it would not be dangerous towards a human, although it is dangerous to an insect. Okay, some plants have kind of different types of things that have been introduced to them. And so some of the things there's strawberry plants that have kind of a natural, from a bacteria, but it's a natural antifreeze. Okay, so this is from bacteria. So it makes it so that if there is a frost or, it gets really cold, the strawberry plants won't die. Both potatoes and rice have been big groups of crops that have been kind of targeted because they feed so many people. So these are very, very important sources of food for many people in the world. And so what they've done is they've actually had kind of potatoes and rice produce harmless proteins derived from cholera. So these are basically, and I'm saying bacterium. So these are kind of edible vaccines. Okay, so vaccinations are difficult to give to people in developing countries. It costs a lot of money, but if you can produce kind of food that would have the edible vaccines in them, expose these usually children to the harmless derived characters of the cholera, then the children will have kind of immunity built up and will allow them to be immune to cholera and not die of it if it happens, okay? There have been other times where people have inserted genes into other kind of food crops that have improved nutritional value. Okay, and so two examples of this are, one of them is called golden rice. It's actually called golden rice 2. And so it carries genes from daffodils, the flower, and corn. And what it's supposed to do is add vitamin A to this food source. Okay, so helps prevent vitamin A deficiency. So people that live in certain areas do not have access to enough vitamin A. And so if you do not have enough vitamin A, it actually leads to blindness. So if you put these genes into a staple food source that can be given to these individuals, then you can help prevent blindness. Golden rice is one of them. And then another food staple is cassava. So cassava is not very common in the United States, but it is a huge food source for many people. Okay, and so it's actually staple for nearly 1 billion people. I think mainly in South America. And so cassava, if it's something that is already being eaten by these people, then if you were able to put genes in there that increase levels of iron and beta carotene, so they've done is increased levels of iron and beta carotene. Sorry, that's a beta. You spell it like this. If you don't, this is kind of the symbol for beta. So beta carotene, that can also be converted to vitamin A. Okay, so again, helping deal with things like blindness. Okay, so there's not many people that kind of argue with this type of thing. As long as it's safe and it's not going to harm these people, you know, why not introduce genes into these food crops that are going to help people live better lives, okay? Simply just by eating their staple foods, okay? Now, not only have plants been genetically modified, but there have been animals. As far as I am aware, there are no animals that are currently you're able to buy for consumption that are genetically modified, okay? However, they have made genetically modified pigs. And those pigs carry a gene whose protein converts less healthy fats to omega-3 fatty acids, which are very healthy. So if you can imagine it, making a healthier version of bacon or any type of pork product, which would be pretty amazing. So one thing I wanted to do, I wanted to see how many people are actually watching these videos. So if you would like some extra credit, I want you to email me and tell me if you would be interested in eating genetically modified bacon. I'm just doing this something stupid so you can get some extra credit points. So email me, I'll give you 2 points of extra credit. Don't tell people. I want people to actually watch the videos. But let me know if you would be interested in eating genetically modified bacon. If you aren't, if you're like a vegan, you can still come in and say like, no, I wouldn't be interested. But wouldn't that be kind of interesting that bacon wouldn't be necessarily as bad as it is for us right now? Okay, so moving on from that. So kind of apart from all of that, you know, agriculture, there is more human health technology. And so one of these that we have not talked about is human gene therapy. Okay, we talked a little bit about it last lecture. We were discussing the kind of the importance of embryonic stem cells and adult stem cells, human gene therapy and things like that. But this idea of human gene therapy is to treat diseases by introducing genes into an afflicted person. So if that person has a disease, they have a mutant gene, you would then be able to give them the good version of the gene and fix whatever disease that they have. Okay, so a mutant gene is replaced or supplemented. So you could maybe you're not going to actually change it in the DNA, but you can somehow supplement the individual with a normal allele Okay. Potentially correcting a genetic disorder. Okay, so if this is a disorder that's because of some faulty gene, so there's been a mutation or something's wrong with the gene, if you could somehow give them the correct version of the gene, that would be unbelievable and would help, cure many, many diseases. There is a lot of kind of promise behind this idea of gene therapy, but unfortunately it has not been as kind of promising, or it's promising, but it hasn't been as, this actual success stories are few and far between at this point, because it's a really difficult thing. And anytime you start to introduce genes into the body, you can potentially start causing damage. But I did want to talk about one kind of success story that has been found. Okay, and it has to do with a blindness that's progressive. And so the blindness is caused by a defective gene. And so what they've done is they've actually inserted the correct version of a gene and put it into a virus. So they do this, it's a different mechanism, but you do a viral injection. Okay, and if you remember, viruses will often insert their DNA into your DNA. And so it can actually carry the gene. Okay, and what happened is that many people that had this had improved vision. And I'm pretty sure they have to inject this directly into your eyeball, if I am correct, but I think it'd be worth it if you had this disease. And sometimes it actually made individuals have almost normal vision. Enough to allow normal vision. So that would be kind of a very big success story, but there aren't a lot of those at this time. But these are the kind of things that, within our lifetimes, hopefully will be more and more successful. The more that we learn about how to do this in a way that it's going to kind of work, and which types of genetic disorders can be helped, which ones can't, and all these different type of mechanisms. So this is something that is very exciting and hopefully will lead to many advances within our lifetimes.